So that moves me into the third part of my talk, which is more specifically about my breeding program at Washington State University. So Washington produces around about 55 to 60% of the US crop. However, the breeding program only started here in 1994. And that's, okay, that's 20 years ago now, but in apple breeding terms, that's still pretty recent. And when you compare it to many of the apple breeding programs around the world, we're still in the juvenile stage. So I have here a schema of my breeding program, and when the comments that I was making about us being in the juvenile phase, 20 years, I think you can probably see here that typically we expect the program to go through about 18 years to produce a variety that's ready for release. So you can see the length of time that it takes to produce a new variety. Certainly one that's thoroughly tested anyway. So I've described previously pollination and then seed collecting. After we've stratified our seeds, we pot them into compost in the greenhouse, and then we can do some testing in the greenhouse for some key diseases, for example, fire blight or powdery mildew. And obviously many people would do apple scab screening at that stage as well. My program uses a rootstock to help us to move through the juvenile phase of the apple tree. If you leave an apple seedling on its own roots, you might be waiting 10 years or more before you get bloom and fruit. And so by using a precocious rootstock, and we use M9 rootstock, we can push through juvenility a little bit faster. The other thing that it does is it produces for us a more consistent tree that is relatively dwarfed, so we don't have to put in a massive amount of effort on pruning, and we can cram a lot of trees into a relatively small space. So my program sends seedlings to the nursery. A commercial nursery works with us, and we get back trees that are on M9 rootstock. We then go through 3 different phases of selection, and I'll talk a little bit more about each of those phases in a bit more detail in a few minutes. We also now have been doing DNA-informed breeding at WSU for a number of years. That first red star at the beginning is to remind me that the most important thing is marker-assisted parent selection, and certainly in terms of my mind when we're talking about DNA-informed breeding. And we've been doing parent selection this way for about 7 years now. And the thing that I really like about it is that it gives you an ability to make a more efficient design for your crosses, because not only then are you taking into account all the phenotypic information that you've got about parents, you also can add in any genotypic information and allelic information that you've got. The second star is the second stage that we've been applying DNA-informed breeding, and that's marker-assisted seedling selection. And we've been doing that now for about 5 years in the program. And this I've been doing in collaboration with my colleague Cameron Peace. He runs WSU's Tree Fruit Genotyping Lab over on the main campus in Pullman. And one of the things that I was really keen to do when I started to bring in this technology into the program was to try and make it as robust as possible, not just with the markers, but how robust could we be, how confident could we be that when we get data back from the lab, we can be pretty sure which seedling it's relating to. It's all a matter of logistics and labeling, and when you're culling a seedling, any decision obviously then is terminal. Once that seedling's gone, it's gone, and you can't come back in a couple of weeks and say, oh, I think that was a mistake, and actually it was seedling A we needed to cull, not seedling B. So one thing that we've done is to develop a system that is relatively robust in terms of our data transfer. We have a sample collection system, which is the photograph you can see on the bottom right-hand side there, with a typical 96-well tube setup. We can sample just a small piece of leaf tissue into these tubes. What we wanted to do was to develop a system where our seedlings are in the same format, and so we have now a 96 plant seedling system that matches our collection system exactly. And so now we can collect straight from the position seedling A1 into the selection tube A1. The other end of this is that when we get our data back from the lab, we also get it in a spreadsheet, which is the 96 well plate format. And so it's very easy then to just stand there with the plate map that you can see there at the bottom with the red and the green squares, and accurately pick out the seedlings that we want to cull. So that has kind of really helped my confidence in our ability to do this testing in the program. So what are we actually testing with? Well, in apple there are a lot of markers that have been identified that are linked to resistances to pests and diseases, but we don't really have very many for fruit quality, and certainly when you go back just a few years, we were really stuck with just a couple of markers, one for fruit skin color and one for ethylene regulation. When you work with something that is a tree fruit, it's really important, I think, to have some of these tools that are linked specifically to the fruit quality, because we have to wait for the fruit. Even using our dwarfing precocious rootstocks, we're waiting 5 or 6 years for the fruit. And that fruit is the most important character. That's really what we're breeding for. And so if we can screen and identify seedlings early on that have those good fruit quality characteristics, then I think that's a really useful move forward in the program. I mentioned that we had these couple of markers, the ethylene regulation there that just shows on the slide the ethylene pathway. Ethylene is a gaseous hormone that is involved in apple ripening, and we have identified a couple of allelic combinations of 2 key genes in that pathway, 2 key enzymes that enable us to select for seedlings that are more likely to store better and to retain their texture. So that's where we were pretty much with fruit quality markers until a couple of years ago. Back in 2009, we got a USDA SCRI, Specialty Crops Research Initiative project funded that was led by Amy Iazzoni at Michigan State University, the RosBREED project. The whole focus of this project was to enable marker-assisted breeding in rosaceae, so not just apples, but a wider range of rosaceous crops. And part of that was to identify more DNA tools to enable us to screen for fruit quality at an early stage. So we spent a lot of time phenotyping and then using a SNP array for genotyping a whole range of different quality traits. And with apple, of course, we're talking fresh traits and also traits after storage. And we're now at the point where we can use some of the DNA tools that we've developed for screening for crispness, juiciness, and acidity. And we're also just at the point of moving into single sugars as well. So I think in terms of fruit quality screening and DNA tools, we've moved on a long way in the last few years. So that's the sort of first part, I guess, pre-phase 1 of the breeding program, the bit before we get to see the fruit on the trees. So phase 1 of my program, this is the first time that we're actually getting seedling trees in the orchard, and we can see fruit. As I mentioned before, these are on M9 rootstock, so they're on the dwarfing precocious rootstock. They're fairly closely spaced. They're about 1 foot apart. We do minimal pruning on these trees, and we have, as I put on there, about 24,000 trees in the ground in this phase at the moment. That's big enough for my program. I don't want to have any more trees in the ground because managing that many is about at our limit. What we do in this phase is select predominantly on appearance and then fruit quality. So my team and myself walk these seedlings usually at least once a week throughout the season. So we're talking, for us, middle of August through to the end of October usually. And then we're really looking for those apples that kind of jump out at us and say, pick me. We certainly do not harvest fruit from all 24,000 trees. Managing these orchards could be problematic, but in fact we have a system of barcoding that I introduced a few years ago now. So each tree has a unique barcoded label. What that enables us to do is when we've picked a fruit sample out in the orchard, we can just when we're out there, we can scan the barcode and print a label immediately that goes onto the fruit sample. This really reduces our error possibilities. One of the worst things that you can do is to go through a number of years of selection or looking at fruit and then go back and propagate from the wrong tree. And it has been done a number of times. So accuracy of labeling is vital. For our fruit samples, all of them go through some cold storage. Typically, we store fruit at just refrigeration temperatures in air storage. We don't change the atmosphere in there at all. And we usually look at this phase of just storing the fruit for 2 months and then putting them out for a week at shelf life, so room temperature shelf life after that. And that's really to kind of just mimic what many people do with their fruit once they've bought it, or at least what happens to the fruit before you actually get to eat it as a consumer. One of the most difficult things with apple seedlings is gauging the right level of maturity to harvest the fruit. My program and many other programs use a starch iodine index as an indication of maturity. And it's pretty simple. You just select an apple off the tree, one that you think is representative of the maturity of the rest of the fruit on that tree. And believe me, within the tree you get a range of maturity as well. You then cut that apple in half, spray on some iodine solution, and then wait for the pattern to emerge, and then try your best to match that pattern with a chart similar to the one that you can see here. This is a chart that Cornell put together, and it's the one that we typically use in my program. We aim to try and pick fruit somewhere between about 3 and 5 on this scale, so we're aiming for 4, but we'll go a little bit either way. Unfortunately, seedling by seedling, you get different patterns of the starch iodine, and so it's very difficult to be accurate on using these indices on seedling trees. And of course, it's destructive. So one thing that we've been looking at is a different measure. How can we find something that improves on this? This DA meter is a meter that's come out of Italy. It was developed initially for use on peaches, but has been used on some apple varieties. We're currently looking at this. What it does is just to measure the fluorescence of the chlorophyll in the flesh just underneath the peel, so it measures through the peel. So it's non-destructive, and you can actually use it, although the photograph shows it there being used in the lab, you can use it on the tree as well. So we're testing this out at the moment, but I'm really hopeful that it's going to work for us, because it would be great to have the opportunity, first of all, to use a non-destructive measure, but also one that I hope will be perhaps a little bit more accurate than the starch iodine index. So once we've harvested our fruit and we've hopefully at the right level of maturity, and we've got it back into the lab and we've done our 2 months of cold storage and our 1 week of shelf life, we do a whole range of different measures of quality. First of all, we do a series of instrumental tests. We use our barcode system all the way through here, and we have the lab set up now so that all of our instruments will just enable us to scan the barcodes in and then dump data straight into Excel, so we don't have to do any of that tortuous data entry and have the sort of errors that you typically get from doing that. This instrument is a more digitest. It's a little bit beyond a simple penetrometer. So in other words, it has a probe. I think you can probably see in that photograph on the left that just pushes into the apple flesh. We usually peel it first, so it's peeled flesh rather than through the peel as well. And this measures the resistance of the flesh, so it gives us an indication of the firmness of the apple, but it also gives us a measure of crispness. And we do this with a 5-fruit sample. We also then take sections of the apples, juice them, again using the barcodes to keep track of everything. And we test the juice for soluble solids content to give us an indication of sweetness. And we also use titratable acidity to measure the acidity of the juice, obviously. And if you choose, you can actually store that juice in the freezer until you're ready to do it. Once we've done our instrumental tests, we go through a whole range of different sensory assessments. We have a team of 4 of us, and we all taste every selection, and then we'll discuss a lot of them as well, because sometimes we disagree, as you'd probably imagine with 4 of us, but I think that's a positive thing. We obviously produce a lot of data, and one of the ways that we've dealt with this is that we've been lucky enough to be able to work with Dorry Main and the team of the genome database for rosaceae. And we have now a database that was specifically designed for my program, so it really helps us to kind of keep track of the data and to be able to compare data from one year to the next. So when we make selections from the first phase, we'll propagate up 5 trees of each of those selections to go into 3 different sites. 2 of these are grower sites, and 1 of them is a WSU orchard. We also put in standard control varieties, and we plant in randomized complete block designs. We have more than 40 advanced selections in this phase at the moment, and because we have bigger trees and more trees, it enables us to do some multiple pick dates. So as I said before, maturity gauging is the most difficult thing. So by doing multiple pick dates, in other words, we'll pick the same sample over a number of weeks, we can hopefully get a better indication of that maturity. And we test the fruit for 4 months as well as 2 months, just because we have some more fruit and we want to push that storage a little bit further. Here you can see the WSU orchard. So the trees are more widely spaced, they're bigger trees, so we'll get more fruit. Because we have more fruit, we can move forward and do some more sensory and consumer work. I work with Carolyn Ross, who's a food scientist at WSU. She has a trained sensory panel that really help us to kind of dissect the sensory traits of some of these selections. And then also, if there's something we're excited about, we'll move forward and do some consumer tests at this phase. So apples that are successful through this advanced stage move into our 3rd phase. Here it's a bigger scale. We're at 75 trees per selection, 4 different grower sites, and all of these grower sites, each grower can use a different orchard system, so the system that they're most comfortable with. And the sites are spread throughout the growing area of central Washington, really to give us a good indication of how well the different selections cope with the different environments. And the whole basis of this particular phase is that it enables us to have a large volume of fruit produced, so much more than we get from phase 2 rather. And I work very closely with some colleagues from the Washington Tree Fruit Research Commission for this phase. They help me manage and keep on top of those orchards, because otherwise it would be a pretty impossible job just from the breeding team. Because we've got larger volumes of fruit, we can do a lot more testing, and storage assessments are key for us. We again do sequential harvest, so we'll pick the fruit over a number of weeks. We do some testing with a chemical that's called 1-MCP, which is an ethylene receptor blocker that the industry uses now a lot on certain varieties to help them to store fruit consistently. So we test samples with and without 1-MCP, and then we also test samples in regular atmosphere storage, so typical cold storage but just in air, and then also in what's called controlled atmosphere storage. So again, it's cold, but the ratio of oxygen to carbon dioxide is controlled. And that, again, enables the fruit to be stored for a longer period of time. So our fruit gets pretty widely tested. We can do this because we work with one of our larger integrated grower packer shippers to Milt in town. They have some storage facilities that they let us use, and we have a very complicated system of samples at this point in time, so it's great to have that space and opportunity. And with a larger volume of fruit, we can do the sort of tests that the industry requires. We can run fruit through commercial packing lines. Some apples fail miserably at this stage, but hopefully we're looking for ones that succeed, obviously. And then we also test waxing, because Washington fruit typically is waxed. And our large volume of fruit enables us to do more consumer tests, and we will do as much as the budget allows at this point in time. You know, we need to get our fruit out there to the consumer and see what they think. So I'm pretty much at the end now. All I just wanted to kind of leave you with is that one of the fun aspects of working with Apple is the diversity that's expected by consumers. And I think that's really nicely coined here in the term the plural nature of perfection. This is something that Howard Moscovich put together when he was describing ketchup, but I think it works really well for Apple as well. When you think about, for example, Granny Smith, Red Delicious, Honeycrisp, any other variety that you can think of, they're all very different, but each has a strong consumer following. So there are consumers that love them and there are consumers that hate them. So for me, I think this whole plural nature of perfection is one of the great challenges of apple breeding. It just leaves me to acknowledge my team, the group here at the WSU, at the Tree Fruit Research and Extension Center, my colleagues at the Tree Fruit Research Commission, and then also colleagues in main campus in Pullman. So thank you very much. I'd be happy to try and answer any questions.